ready, so please go for it.
Visible. Yeah.
Yeah, we see your cursor.
Okay. Good. Okay. Hi. So today I would like to talk about a recent result we have published in Physical Review Letters, where we present an analogy between, on the one hand, the durotactic guidance of nerves, which I will detail in a minute, and on the other hand, the classic rules that govern classic ray optics, hence this rather cryptical title, An Optic Ray Theory for Nerve Durotaxis. So this is a work that I did with Alain, the Mathematical Institute of Oxford, in collaboration with Christian. So during the establishment of the neuronal system, neuronal cells extend processes which are called neurites. And during this phase, one of the neurites will come to dominate the others and differentiate in an axon to connect to distant functional targets. And the activity of the growth cone, which is the tip of the axon, which is an actin-enriched structure that performs both sensory and locomotory functions of the axon. So what happens is the growth cone produces forces and pulls itself on the medium, which results in elongating the axon, the trailing axon shaft behind it. And in this phase, the growth cone, in order to find its targets, relies on different kind of signaling pathways, most important of which being, of course, the chemical pathway, and in particular chemotaxis. But it also depends on lesser known mechanisms such as mechanical environments, and in particular durotaxis. So durotaxis, generally speaking, refers to the directed motion of cells along a gradient in medium stiffness. What you see on the right side of my screen is the experiment that is at the genesis of this work. So this is an experiment that was conducted by in Christian's group, showing the left part of a xenopus frog brain, and the red thing you see on the screen is the optic tract that extends from the right retina, which is not represented here, to the optic tectum in order to form the visual system of the frog. And what is interesting on this trajectory is this very stereotypical turn made by the optic tract on its way to the tectum. And we know that this turn depends on mechanisms like chemotaxis, and it involves, in particular, chemorepellents like slits. But what Christian's group have found is also the correlated emergence of a very strong gradient in stiffness between both parts of the bundle, where the front part is approximately 10 times stiffer than the rear part. So what they did is, in order to test the potential role of this gradient, this stiffness gradient on the trajectory, is grow axons on different kinds of substrate with different stiffness in vitro. And of course, I'm skipping a lot of details, but what they observed is that axons tended to grow faster and straighter on stiffer substrate. And so by mapping this in vitro results onto the in vivo scenario, what they proposed is that perhaps the axons growing on the soft part could act as an anchor to the whole bundle, causing this deflection of the bundle. Okay. So in order to gain a more rational understanding of this process, Alain and I started to develop a model for this phenomenon. So in this model, we assumed that all axons were tightly coupled mechanically. And this can be represented using a rod model. And the tip of the rod represents the front of the bundle made up with all the growth cone that pull with different force on the substrate. And in particular, the important general effect is that the force increases with the local substrate rigidity. For those interested in the equations, the starting point of this model consists in expressing the dependency between the force produced by each growth cone on the substrate rigidity. So simply we assume that every growth cone in the bundle pulls with a different force and in a straight line. And we assume that this force is proportional to the substrate stiffness, which allows us to model the durotaxis effects. And now if we integrate all these effects, we obtain a global torque, which is applied to the bundle front, which is obtained by summing all the infinitesimal torques produced by each growth cone, which results from the fact that each growth cone is located at a certain distance from the central axis of the bundle. And last, if we want to obtain an actual trajectory of the bundle, which is a geometrical notion, we need to express how much curvature is created by a given torque, which is something that can be described by introducing the notion of bending rigidity, which is the product of the Young's modulus by the second moment of cross-sectional area. And so these equations are very general and apply to any kind of substrate stiffness. And so to understand better the kind of behavior that can emerge from this model, we studied the canonical scenario of a bundle crossing an interface between two zones with different stiffness, so two zones with different uniform stiffness. And if we make this assumption, we see that the set of equations I just showed you boil down to this very nice nonlinear dynamical system that depends in particular on a single non-dimensional parameter, the durotaxis number, which is proportional to the jump in stiffness between the two zones. So to gain a geometrical understanding of what's going on, a classic thing that is done in applied mathematics is what is called phase portrait analysis, and represents a diagram where every position represents a possible configuration of the bundle. Configuration meaning a position and an orientation. And from every position, we draw a little arrow which represents the immediate direction that the system will take from a given position. And what we see here is a clear white almond-shaped region where deflection occurs. And this corresponds to the only portion of the domain where the bundle actually interacts with the interface. And outside this domain, the bundle grows on the same zone. So every growth cone is subject to the same stiffness field and grows in a straight line. So if we start from a given angle and we shoot the bundle to the interface, we see that the trajectory in the phase plane makes this kind of U-turn around this fixed point and exits the interface from the same side, which in the physical space resembles a reflection regime where the axon sort of bounces against the interface. If we now choose a slightly different angle and an angle which is closer to the normal to the interface, we see a radically different regime where the bundle, instead of being reflected, makes it through the interface and exits the interface with a different angle, which is of course very reminiscent of what is observed in classic optics with reflection and refraction of light rays. And as a matter of fact, the next part of this work consisted in pushing this analogy analytically and try and understand whether to what extent this analogy was accurate. So I mentioned the laws of optical refraction. Those are well summarized by Snell's law, that governs light refraction at an interface. So Snell's law is named after Dutch physicist Willebrod Snellius, who proposed this law, although French prefer to give the credit to Descartes. And there was actually, during the 7th century, a little debate on the parentality of this law. And we can mention also the contribution of Pierre de Fermat, who also proposed an approach that had momentous effect on the development of analytical physics. But anyway, it turns out that this law was known since the 10th century and was proposed by IBMTL in Baghdad 7 centuries ago then. So anyway, what Snell's law tells us is that if we send a light ray to a refractive interface, so an interface between two zones with different refractive indices. So the refractive index, roughly speaking, quantifies the optical density of the medium. So this light ray can behave in two different ways. It can be totally reflected, is the angle with respect to the normal is large enough, or it can be refracted with, and by doing so, it obeys this law that I've written here called Snell's law, that says that n1 sine theta 1 is equal to n2 sine theta 2. And if we stretch a bit the analytical development from our model, it turns out that quite miraculously, it can be proven that our equations, in the case of a straight interface, behave in the same way as an optic ray following Snell's law. And this is quite a nice result, because it means that potentially all the nice results that result from Snell's law and all the nice applications of Snell's law, could find potentially a counterpart in the case of durotaxis, and this is what I'm going to demonstrate next. One of the obvious potential effects is what we call durotactic lensing, which can be observed here by putting a stiff obstacle in front of the axon path. So what you see here is two types of line. The solid line represents the trajectories that are obtained by direct integration of our nonlinear system, and the dashed line are obtained by doing the Snell-like construction based by assembling piecewise linear functions. And conversely, we can also obtain convergent lens by inverting the two zones and considering a soft obstacle. Another problem we looked at is actually a problem which is quite essential to the problem of guidance, is how to guide axons from a point A to point B with minimal loss. And it turns out that this is an important problem also in light-based communication technologies, and people have come up with elegant solutions, in particular the optical fiber, which is a refractive tube that allows to trap the light by exploiting total internal reflection and trap the path of the light between the two. And so we adapted this idea by proposing this sort of a virtual experiment consisting in growing a bundle of axons between two stiff zones and trapping and forcing the axon to grow between these stiff zones in a soft corridor. This is a simulation of this principle where we have I have represented two trajectories that are subjected to the same subject to the same noise. So we represent stochastic effects that can cause a bundle to get lost, and we can see that the durotactic effects allows to trap the bundle between the two zones and reach the target if we consider that the right part of the plane is the target. And the last thing we did, I would like to go back to the example of the Xenopus visual system, is try the system on the stiffness map obtained by atomic force microscopy, which we extracted from the paper by Thompson et al. In 2019. And by using this map, we could simulate our model very easily, and adding some noise on the trajectory and the initial position. And we managed to, and this shows that the durotactic effect has indeed the potential to contribute to this caudal turn. Although we cannot really see the sort of sharp angle that we see in vivo. So which suggests that potentially other effects like chemotaxis, which is actually a known effect, and steric hindrance by cell bodies could also have a very important effect on this process. So I have presented a minimal model of axon bundle where we have represented the axon bundle using a tip growing rod model, which is a very easy model which allows to perform analytical development which allowed us to propose a simple law governing the deflection of axons by a stiffness interface. And it turned out that, quite surprisingly, this law was equivalent to the law that governs light refraction at a refractive interface, that is Snell's law. And I would like to stress, in case this was perhaps not very clear, that this is not a contrived result that we may have wanted to force into our model from the beginning. And as a matter of fact, we were quite surprised to see that this sort of optical effect emerged from assumptions that are in nature predicated upon very fundamental mechanical considerations that have seemingly nothing to do with light propagation physics. We see this kind of prediction as quite powerful potentially, as it could help building a simple frame of understanding for durotactic guidance and maybe guidance in general. And to finish, I would like to say that as applied mathematicians, we are, of course, very sensitive and convinced of the potential intellectual and pedagogical power of making parallels between physics and biology. To finish, I have put the reference to the paper in Physical Review Letters, which was published last month. I would like to thank my collaborators, Alain and Christian, who are co-author on the paper, and also Ryan Greenhalgh, who is a young DPhil student in Cambridge who helped me understand the problem. Thank you very much. And if there is any question, I would be happy to take them.
Fantastic. Thank you very much, Hadrien. Great talk. I'm happy to take questions from either Q&A at